Okay, so certain bacteria will require special conditions to grow. For example, anaerobic bacteria. If you remember this from earlier, these bacteria which die or fail to grow in the presence of oxygen. So anaerobic jars, for example, the gas pack system, can be used to culture a small number of anaerobic bacteria. And in this system, inoculated plates, so the plates with the bacteria, are placed inside the jar, along with the gas generator sachet, the gas pack, and an indicator strip, and then the jar is sealed completely. And then in the presence of water, the chemicals inside the gas pack react to produce hydrogen and carbon dioxide gas. The hydrogen produced then reacts with the oxygen present inside the jar, producing water, and this forms as condensation on the inside of the jar. And in this way, the oxygen is removed. So as the oxygen is removed, the indicator strip changes from blue to colourless. Okay, so this will show that the system's working. Just a note as well that newer gas generator systems are now available, which actually don't require the catalyst and don't require addition of water to these systems. So anaerobic chambers can also be used to culture anaerobic bacteria on a larger scale. And this chamber works by removing the air with a vacuum pump, followed by purges with nitrogen gas. So the nitrogen gas pushes the air out of the chamber. A gas mix containing hydrogen is then introduced into the chamber. And then in the presence of the catalyst, the hydrogen and any remaining oxygen react to form water and hence remove the oxygen from the chamber. Okay, so as we mentioned, different bacteria require oxygen to a different extent. And the nature of a bacteria's relationship with oxygen can be easily determined by growing the bacteria in a medium such as thioglycolate broth. And this contains a reducing agent to reduce oxygen levels. So when grown in test tubes with thioglycolate, the obligate aerobes, which are dependent on oxygen for growth, gather at the top of the tube where the concentration is highest. The facultative anaerobes can grow with or without oxygen, but grow better in its presence, so they gather mostly at the top of the tube. And then you have the aerotolerant anaerobes, which grow equally well whether oxygen is present or not, so they are found evenly spread through the test tube. Then you have the obligate anaerobes, so the strict anaerobes, which cannot grow in the presence of oxygen, so these gather at the bottom of the tube where the oxygen concentration is lowest. And then you have the microaerophiles, which are damaged by high concentrations of oxygen. So they gather in the upper part of the test tube, but not at the very top. So the next part of the lecture will look at the phases of bacterial growth. But before we look at this in more detail, we will revisit how bacteria divide and reproduce. So we have discussed previously how most bacteria divide by binary fission. And this is a continuous process as long as the environment permits. For example, they don't run out of nutrients. And the time taken for bacteria to double is called generation time. And this generation time varies a lot with different species and environmental conditions. So whereas most pathogens grow rapidly, there are exceptions to this. So for example, mycobacteria are known to grow very slowly. And Mycobacterium leprae, which causes leprosy, has a doubling time, which is quite long, of 14 days. So if we look at the phase of bacterial growth in more detail, so the first phase of growth is called the lag phase, and this is when a small number of bacterial cells the inoculum, is added to fresh culture medium. And in this lag phase, cells do not start to grow and divide straight away. Instead, in this phase, the cells are gearing up for growth. So they are synthesizing new cell materials, for example, proteins, ATP, and carrying out any repairs that need to take place. For example, if the cells have been injured during the transfer to new medium. Eventually, however, the cells will begin to replicate their DNA. They'll increase in mass and then their numbers and divide, and their numbers will start to increase. And the next phase of cell growth is the exponential phase. And this is where the bacteria grow and divide at the maximum rate possible. So the growth rate is constant and the bacteria cells are doubling in number at regular intervals. And when conditions are optimal for the cells, this will result in a steeper slope in the growth curve. This stage of bacterial growth is ideal for biochemical and physiological studies because the culture is chemically and physiologically uniform. So it's very useful when doing experiments for all the cells to be uniform. So in a closed system such as batch culture, this exponential growth cannot last forever. So eventually the growth will start to stall due to several factors, including the depletion of nutrients and oxygen. You can see how the growth curve then becomes horizontal, and this is when we're in the stationary phase, where the total number of live cells remains constant. And this may be due to a balance between bacterial cell division and cell death, or it could just be that the population may just stop dividing but remain metabolically active. Okay, so the last phase of the growth curve is the death phase. So cells growing in batch culture will not stay in the stationary phase. They will eventually enter the death phase. And during this phase, culture medium accumulates toxic waste and the nutrients have been completely depleted. And the number of live cells decrease exponentially, with cells dying at a constant rate. It has been shown, however, that 100% cell death is unlikely for a population, as the cells mutate and adapt to their environmental condition. So often there's a tail-in effect observed, where a small population of the cells cannot be killed. In addition, these cells may benefit from the death of the fellow cells, which provide nutrients to the environment when they lyse and release their content.